Hello, good morning, everyone. Today we're going to be doing lecture 7, which is about protein synthesis. All right, so we're going to talk about how we will be taking the genetic material to DNA, and then we translate it into RNA. And then from the RNA, we're going to go and produce the protein. So the entire process is kind of, a little bit complicated, but hopefully I'm going to help you understand it better after the tutorial. Okay, so let's get started. First question, what is the central dogma of cell biology? We talked about this before, but it's a good idea to refresh our memory before we discuss the protein synthesis process, because essentially the central dogma is surrounding it's a whole theme that surrounds the process of protein synthesis. So we start with the DNA, which is in the nucleus. Okay, and we're going to make a copy of the DNA in the form of RNA. Okay, so that process is called transcription. Okay, remember, DNA and RNA is kind of the same language, right? They both use nucleotides as their coding method, and that's why this is called transcription. Okay, and again, this happens in the nucleus. Now, once you make the RNA, you will have to modify it, and we're going to talk about those modifications later on. But then it will exit into the cytoplasm through the nuclear pores, and once it's in the cytoplasm, it is going to be translated. Okay, it's called translation because DNA, RNA is the same language, nucleotides, but RNA and proteins, they are different language, right? Proteins is encoded using amino acids, whereas RNA is nucleotide. And this process happens in the cytoplasm. Okay. Next one, we compare the DNA and RNA on multiple occasions, but again, it's a good idea to refresh our memory on that. For DNA, it is going to be double-stranded. Okay, whereas RNA, it is single-strand. Now, even though RNA is single-stranded, they can fold back on themselves. So let's say I have a strand of RNA like this, okay? And what happens is it's possible for it to fold back on itself to form various structures. Okay, so like this. So for example, this part of the RNA might be a ACCU, and then this part might be a TGGA, right? So that way, I'm just zooming here for you. So that way it's able to form these complementary bonding with itself, and it folds up into these various shapes. If you watch the lecture recording, then you would know this is what like a transfer RNA would look like. Okay, so even though they're single-stranded, it doesn't necessarily mean that they have to exist as a long linear strand on its own. It's very often that they fold up into various structures. Type of ribose sugar. For DNA, it is a deoxyribose. Okay, it's missing an oxygen. You should probably know that by now. Whereas RNA is just a regular ribose. Nitrogenous base, ACTG for DNA, but for RNA is ACUG. And you have the uracil right here. Different types of DNA. We have just one type, it's just DNA. Whereas RNA, what are the types of RNA that we have? Anybody can help me out here? Yep, Tyler said there's messenger RNA, very good mRNA. Anything else? Domain say tRNA, yep. And Tina said rRNA. They are all correct. So there's 3 types of RNAs. And actually, these are the main ones that we will be talking about. There are other forms of RNA that exist in the body. That is quite cool. Okay, so this one you don't have to know, but I just feel like talking about it. There's something called siRNA. Okay, it's called silencing RNA. And what happens is the silencing RNA can bind to a certain gene, and it will cause it to turn off. Okay, so this is an area of very intense research, because if we can create these silencing RNA, we will have a way to turn off certain genes, perhaps in like a cancerous cell. We can use the silencing RNA to turn off the gene that causes it to have the ability to keep on replicating. For example, we talk about how in cancer cells, there is an elevation of telomerase last class, right? So what if we are able to use this silencing RNA to turn off that gene so that it's no longer going to be transcribed or translate, and therefore control the growth of the tumor, right? So that again is something that researchers are doing. Anyways, moving on, 3 types of RNA and their main function, right? So we already listed them out, but let's write down what is it that they do. Right? So the first one is the messenger RNA. I probably should write out the name, okay? mRNA. So the M stands for messenger. Okay, so essentially what this is it is a copy of a gene which contains the instruction to make a protein. Okay, so proteins, as you know, they are amino acids, right? You have to link them together one by one. But how do you know which one to link together? How do I know to put the phenylalanine next to the alanine and so on and so forth, right? That turns out, that instruction turns out to be encoded within the DNA, but we cannot take the DNA out of the nucleus. It's too big. So that's why we're gonna have to, just copy what we need and take it out to the cytoplasm in the form of the messenger RNA. So in a way, this is like the recipe, okay, to make the proteins. Okay. Next, we have the, doesn't really matter. We can do it in the same order here. We have the tRNA. Okay. The T stands for transfer. Transfer. Okay. So transfer RNA is responsible for bringing, so it brings the correct amino acid to the ribosome. Okay. So the recipe tells you what amino acid to link together, but something has to bring that amino acid over, right? Okay. So if we need a phenylalanine, the tRNA carrying the phenylalanine will come over and give it to the ribosome, and so on and so forth. Okay. And also, right, it's important to know that at the bottom of the tRNA, okay, the tRNA contains what we call anticodons, and I'll explain what that means later if you don't know. Contains anticodon that is complementary to codon on messenger RNA. Okay. Finally, we have rRNA. Okay. And the R stands for ribosomal. Okay. And excuse me, sorry. And the ribosomal RNA is a part of the ribosome. Okay. The ribosome is made up of the ribosomal RNA as well as proteins. Okay, and the function of the rRNA, partly anyways, is to actually synthesize the protein, right? So synthesize protein. Okay. So again, mRNA is like the recipe to make something. The rRNA would be the chef that's actually making it. And then the tRNA would be the helper that's bringing all the ingredients necessary to make the dish, the protein. Okay. Any questions so far? Okay. Next, we have list the 3 steps of transcription. The 3 steps of transcriptions are initiation, followed by elongation, and then termination. Now, just to write down very briefly, I'll call this 1, 2, 3. So number 1, initiation involves the binding of RNA polymerase. At this time is RNA polymerase because we're building RNA, right? Last time we were making DNA, so that's why it was DNA polymerase during DNA replication. But for transcription, we need RNA polymerase. So binding of RNA polymerase to the promoter of a gene. Okay. So the promoter is a special sequence that marks the beginning of a gene. Probably should write it down for you. There's a lot of, just like a little footnote here. An AT-rich region that marks the beginning of a gene. Can someone tell me why it is AT-rich, please? Why is a promoter rich in AT? That means like it has a lot of A's and T's. Why is that? No? That's okay. So in order to make a copy of a DNA, we have to unwind it, right? We have to unzip it. So by having more A's and T's is easier to pull them apart, okay? Because remember, between A and T, there are only 2 hydrogen bonds, right? Whereas C and G, it has a lot more hydrogen bonds, right? Okay, so we want to have a region that is easier to pull apart so that we can slide the RNA polymerase in and begin the process of transcription. Okay, I think on the test bank there is a. Do I have that question? I don't have that question here with me. But then on the test bank there is going to be a question that asks you to identify the promoter. I'm going to give you a sequence, right? And ask you to identify the promoter. Okay, so you would just choose the one that has a lot of consecutive, well, not consecutive, but then you would have a lot of A's and T's in it. Okay, number 2. Number 2 is elongation. Okay, so using the template strand, right? RNA polymerase will build a complementary strand of RNA. Okay, and finally, termination. Okay, RNA polymerase falls off when it reaches the terminator. Okay, so terminator marks the end of a gene. Is this okay? I mean, this is a very brief summary. But does anyone need any clarification? All right. Okay, let's go over here. Draw and label various parts of pre-mRNA, pRNA, and the processed messenger RNA. Okay. Now, once you go through this transcription process, the RNA that you get, it's not the final product. Right? So we have, let's say this is like we will call this the pre-RNA. Okay, pre-RNA. And on the pre-RNA, there are some parts that we need to remove. Okay. So these are introns. Oops, I need a thinner pen. Introns. Okay, so we have intron here, intron, and intron. So these needs to be removed. Okay, whereas these ones, right, we will call them exons. Those are the ones that will be expressed. Exon, exon. Okay. So these are the coding part of the gene. Whereas the introns, they are the non-coding portion. Okay, non-coding portion or parts. Okay, so we need to remove them. So one of the things that happens in post-transcriptional modification pro-transcriptional modification is that we will undergo RNA splicing. Okay, so in RNA splicing, we will remove introns and connect exons. All right. Now this process is actually quite interesting. We'll revisit it in next semester because you don't actually have to take out all the introns. Sometimes you can choose to take out intron number 1 and number 2, but keep intron number 3, or you can choose to take out 2 and 3, or all of them, or just the middle one. Okay, so by deciding which one to remove, you actually can create variations. Okay, and the ability to create variations like that is very important to generate things like antibodies. And we'll revisit that next semester. Okay, what else is going to happen besides RNA splicing? Okay, what's another thing that we have to do in post-transcriptional modification? Help me out here, please. What do we have to add? Does anybody remember? Yeah, thank you, Tyler. So number 2, we have to add a 5' cap. Okay, 5' G cap. Okay, it's guanine based on the nucleotide guanine. So we call it 5' guanosine cap. And then number 3 here, we add a poly-A tail. Okay, poly-A tail. The longer the tail, the longer the half-life in the cytoplasm. Okay, so if something has a really long poly-A tail, you can keep on making that protein for some time. But if it's really short, it will break down after a few rounds, and it's just another way of controlling how much of the proteins you're going to make. All right. So that is that. Okay, now we're going to have to talk about the actual transcription process. If I give you a strand, you have to be able to come up with all the other stuff. All right. So let me just talk about the difference between coding strand and template strand, because it is something that people actually get confused easily. So here we have the RNA polymerase. That's what this gray, guitar pick looking thing is. This is the RNA polymerase, and the blue strands here are the DNA. Right. So we are looking at the elongation process, and you can see the top strand here is the coding strand. The coding strand is the strand of a DNA that contains the actual genetical for making a protein. Okay. Instead of using the coding strand to make the RNA polymerase will actually be using the template strand to make the RNA, okay? Because of the way of how the polymerase works, right? That it only knows how to build things using complementary base pairing rule. You have to use the template strand. Okay, so you can see the coding strand and template strand are complementary themselves, right? We have T and A and G here, and so T is complementary to A is complementary to T, G and C, so on and so forth. So what happens is when RNA polymerase use the template strand to build the RNA, right, the end product, which is shown here in red, is going to be identical to the coding strand. So you see the red thing that you end up building is exactly the same as the coding strand, right? With the exception that U replaces T because the RNA has no thymine on it, right? So we have to replace it with uracil. But other than that, it's the exact same thing. So C, A, U replaces T, and then C, A, so on and so forth, right? So that's why if you are given the coding strand, the mRNA that you create will be identical to the coding strand, with the exception that T is replaced by U. If you are given the template strand, however, when you make the mRNA, you have to use the complementary base pairing rule. And so let me summarize that for you one more time. Okay. So the messenger RNA, or the RNA, would you say? We can say messenger RNA, doesn't really matter. The messenger RNA, right, the red one here, is complementary to the template strand. Okay, meaning if the template is ACTG, right, template, then the mRNA is going to be UGAC. Okay, so that's number one. Now, number two, the mRNA is identical to the coding strand. Okay, the coding is the lighter one, right? Except U replaces T. So if I have a coding here of ACTG, and the mRNA, it's going to be the exact same thing, but you replace the T with a U. Okay, this is the key thing that you have to remember when you do those transcription questions. Okay, so give you a moment to copy it if you need to. And also, does anyone have any question about this? Okay, so let's do it. Over here, we are given the coding strand. Okay, so the coding strand is going to be, as I mentioned, identical to your mRNA, right? Okay. So I'm going to copy it. ATG, it is going to be AUG. Okay, I must replace the T's with U. Replace T's with U's. So AUA, CGA, AAU, GAU, UAA. Does that make sense? Does anybody not understand how this is done? It's important you know how to do this because a lot of questions will be revolving around this concept, right? Okay. Now, anticodon. We mentioned anticodon earlier. It is found in the tRNA. Okay, so let me just draw the messenger RNA here again. Right, we will have the AUG, AUA, CGA, and then the AAU, and then so on and so forth. Right. So we will have the tRNA right here. Okay, tRNA. And at the bottom of tRNA, there is going to be an anticodon. Okay, that will be complementary to the codon right here. So what is complementary to AUG? Can someone tell me what's complementary to AUG, please? Yep, it is UAC. Okay, UAC. And not TAC because this is also RNA, right? So it will only have uracil and not thymine. Okay, I hope that makes sense. And so if these things are complementary, then this tRNA will be allowed to come and bring the correct amino acid. Okay, but what amino acid is it bringing? Well, if the codon, and I should probably label it for you, this is the codon and this is the anticodon. Okay, so what does AUG encode for? Well, that's when you're going to need to have the codon chart. All right. So this is the one that I post. Well, this is the one I use in lecture, the one on the left. But then the one on the right is the one I post on eCentennial under the announcement. I think this one might be a little bit easier to use because it tells you exactly what the amino acid is going to be. So the codon we are looking for is AUG, right? Okay, so we start with the first one, which is A. And then the second one is U. Right? So this row is for all the A's, this column is for all the U, and where they intersect is what we're interested in. Okay, so we're looking for AUG, and you can see AUG codes for methionine, okay, or MET for short. Right? So that means when they come back to here, right, this code is asking tRNA to bring the methionine. Right? So that's why the tRNA with the complementary anticodon carrying the MET will come along and deposit the methionine. Okay. If I have another tRNA here, okay, that's carrying something else like PHE or something, the anticodon at the bottom is not going to be complementary to this codon, and therefore it won't be able to dock, right? So it's not going to be bringing phenylalanine. Okay? So only the amino acid that you need will be able to come because of the complementary pairing. Okay. So why don't we write down all the anticodons here for a second? Okay. So it's going to be complementary to the mRNA, as I said. So this would be UAC, right? UAU, GCU, UUA, CUA, AUU. Okay? Just check that over. Make sure you understand it. Right, so these are complementary. All right, now we're gonna look up the I'm gonna look up the amino acids, right? And let me pull the codon chart over so we can do it side by side. I'm not sure how this is gonna work. It's the screen is not big enough, but hopefully You can still see. All right, so the first one is we're looking at the mRNA, right? Okay, so AUG, we did that already. It was MET. Okay, MET. Okay, next one, AUA. Okay, what does AUA code for? Please tell me what AUA codes for. Okay. Does everybody agree that it is going to be isoleucine or ILE? That's I know it looks like LLE, but it's actually ILE. It's one of the strange things. Capital I looks exactly the same as lowercase l on the computer, so sometimes it's hard to tell. But it is ILE. Now, how do you do that? Okay, if you're not sure, again, watch. We look for A, which is right here. That's the first one. U is the second one. And then this is where the two intersect, right? And then we're looking for AUA. So AUA is right here. That is ILE. You do not have to know the full name. Okay? You can just know the short form. That's good enough. All right. So I'll do one more with you, and then I'll just write out the answer. Okay. CGA. So C is here. G is here. So CGA is arginine, ARG. Okay, and if you keep on going, you will be able to find all the answers. Yeah, and finally, when you get to the end, you have UAA. So UAA is one of the 3 stop codons, right? Yeah, so the process of translation is going to stop. All right. There are 3 stop codons. We have UAA, UAG, and UGA, right? Okay, and in the slide I told you to memorize them, you can think of the acronyms, right? Like you got A in biology, you're good, you're awesome, right? So UGA, UAG, and UAA. Okay, let's try the next one. Go ahead, try that. I'll take it up in a second. Now, this is the template strand. So what's the relationship between template strand and mRNA? We talked about that earlier, right? It is going to be complementary. Okay, but again, of course, right? Anytime you need to have an A, you will use a U instead. Anytime you have a A, it will pair with a U instead of a T, right? So over here, what is the strand? Okay, I'll write out for you. And if it doesn't make sense, please let me know. Okay, so TAC will be AUG. Okay, CCC. GCG, UAA. Okay. And the anticodon will be complementary to the mRNA. Now, I don't know if this is going to confuse you guys, but for some of you, you will notice that the anticodon and template strand are actually the same. Right? These two things are actually same. Right? But T is replaced by U. Okay. So I guess we can do this. We can do this. Okay. We can we can turn on like cheat mode, if you would. And you can put this on a sticky tag and have it ready for you on the test. So we have template, we have coding, we have mRNA, and anticodon. Okay, so template, whatever, TACTG. Yeah, there's only four possibilities, right? Like you cannot really have anything else. Okay, so ACTG. On the coding strand, it would be, okay, help me out here. Okay, I only see a few people answering. Are the rest of you with me? What would the sequence be? If the template is ACTG, what would it be for the coding? That's right, TGAC. I hope that makes sense, okay, because they're complementary, right? And then what about the mRNA? Well, the mRNA is going to be UGAC, right? And the anticodon will be ACUG. That's it. Okay, this is the cheat sheet. Put that on a sticky tag. Okay, stick it next to your monitor, because that's what you need to do these questions. Okay, so how to use this? Look. Template is TAC, right? So you look at the relationship between template and anticodon, for example. You can see they're the same, but the U replaces the T. Okay, so this is probably the most important thing to write down. I hope that helps. Okay, amino acid. You can just look it up again, right? On the chart. Does everyone understand how to use this chart? Anyone need me to go through this again? You can private message me if you don't feel comfortable you know, saying it in the public chat. Does anybody want me to talk about this again? Because if you don't know how to use this chat, you're not going to be able to do the test questions. We are good. Okay, I'm trusting you. Okay, so this is MET Pro. Okay, I'm just copying from my previous answer key. That's why I'm doing it so fast. And then stop. That's it. Okay. So if you get something different, please do let me know. Okay, we'll do one more. Here we are given the mRNA and you are asked to find a template strand, right? So again, if you are confused, you can just look at the cheat sheet here. Okay, so if I have mRNA and I need to find a template strand, right? Let's see the relationship. It looks like they're complementary. So over here, we will have, instead of AUG, we will have TAC, okay? And then TGG. Remember, we have T now because this is DNA, right? GTA, CGC, AAA, and ATC. All right. As for the anticodon, we will have, what are we going to have? We're going to have UAC, UGG, GUA, CGC, AAA, and AUC. Okay. This is either very exciting for you because it's simple or it's very boring. Okay. Depends how you look at it. And then you will just look it up like before. Okay. So I'll leave this part for you to do on your own. Okay, let's do some poll questions. Let's see what we have. Oh, this one is about deletion. We'll come back to it later. Oh, this is a good one. Let's try this one. Go ahead. All right, I have 2 responses and it has been a minute and a half. Where is everybody? Oh dear. All right, I'm gonna call it. Anyone else want to lock in their answers? Even if you don't know what the right answer is, if you see this question on the test and this question will be on your test, what are you gonna choose? What does your gut feeling tell you? All right. So thank you for the 8 of you who have participated. Okay, well, a regular codon has 3 nucleotides, right? So, you know, the regular has 3 codons, 3 nucleotides. So how many combinations are there? In other words, if you look at this codon chart, how many codes are there in total, right? Okay, so you can count them all if you want, right? Or you can, you know, use math. And if you use math, you will know that the first slot, how many choices do we have? Well, it can be A, U, C, or G. So there are 4 possibilities. Same thing for the second one, and same thing for the third one. So if you multiply them all together, 4 times 4 times 4, that gives you 64, right? Very good, Adriana. Now they're saying, the question is that what if we have 4 slots, 4 nucleotides instead of 3? Well, it's just going to be 4 times 4 times 4 times 4, right? Okay. So what does that work out to be? So that's, you know, 64 times another 4, which is 256. Is that okay? Okay. All right, let's try this one. This one should be quite straightforward. Okay. And looks like all of you got the answer, right? Okay, so this is the template. So rather than looking for the answer, you can probably just write it out first yourself, and then you match them up, right? Okay, and so this is it. Okay, good. All right. Now, before we go on, I just want to you know, mention something. As you probably know by now, that you know, unfortunately, there might be a strike on Friday if you know, if there's no negotiation going on, right? And so I don't know what will happen next week. But we have the quiz that was supposed to happen next Tuesday, right? Okay. So again, I don't know what happens if there's a strike. Then I won't be able to access the homepage and all that stuff, and you won't be able to contact me. So what I did is that I opened the quiz. Next week's quiz, I opened it today already. Okay, so for those of you who wish to do the quiz before the end of the week, you have the option of doing that. Okay, so that way, if you run into any problem, you can still contact me. You don't have to do it, right? If you feel like you want to do it next week, that is still fine, I think. But you know, I just want to minimize the amount of stress that you guys, you know, have because of this. So yeah, Okay, it will be available from today all the way until next Tuesday. Okay, so I hope that will help a little bit. And I understand it is very difficult for for you guys to be put in a situation like this, and for that I truly apologize. But yeah, let's hope we won't have to actually go on strike. Okay, but who knows? Okay, and thank you for all the kind messages in the chat box. Okay, let's move on back to this thing. Number 7. Okay, given the following mRNA sequence, which 2 codes for the same protein? Now, this is tedious and time consuming, really more than anything else. All you have to do is translate these sequences and see which one has the same. Okay, so you will look up AGU, which you use the codon chart, right? A, G, and U, which is SCR, right? So you write it out one by one, okay? And I'm not going to do it right now because it's not the best use of our time. But when you do that, right, for all 3 of the sequences, you will find out that this and this are the same, okay? The first and the last one. Okay, next one, also a good question. If a DNA sequence contains 360 nucleotides, how many amino acids do you expect to be in the protein sequence it encodes? Okay, what do you guys think? How many? Yeah, 120 is correct, right? Because 360 divided by 3. Right? Why divided by 3? Because there's 3 nucleotides in the codon, right? So if you divide by 3, then you will get 120 amino acids. Okay, assuming there are no introns in the sequence. Okay. So I guess in the test, I can say instead of putting you to guess, I might say what is the maximum number of amino acid that would be encoded by this. Okay, so the maximum would be 120 amino acids. All right, next we have all the mutations. Okay, the mutations. Okay, so over here, we have the normal mRNA strand, and we have all the amino acids here, and on the left side here we have what's called base pair substitution. Okay, so you're replacing the nucleotides with something else. Okay, so over here, if we compare the first one to the original one. By the way, the normal RNA, that is also called the wild type. Wild type. Okay, wild type sequence. And if you compare them, you can see there is a change over here. Instead of ACA, it has been mutated to ACU. Okay. But fortunately, if you look up the codon ACA, if you have the codon chart next to you, you'll see that the ACA codes for threonine, okay, or THR. And ACU also codes for the same amino acid. Okay, so because of that, there's actually no change in your amino acid sequence despite the changes in the DNA sequence. Okay, so something like this, right? It is called a silent mutation or a neutral mutation. Okay, and this is because ACA becomes ACU, but they both codes for the same amino acid. All right. So if you compare the DNA of one person to another person, there's going to be a lot of differences, right? Okay, and a lot of these differences are basically these silent or neutral mutations in the body. Okay, we call them SNPs. Okay? We'll revisit the idea of SNPs later on in lecture 9, the biotechnology. So this is a single, for those of you who are interested, single nucleotide polymorphism. Okay, polymorphism is basically the super fancy way of saying different. Okay, so SNPs are differences based on a single nucleotide. Okay, so for you, you might have ACA in your genetic code. For me, I would have like ACT technically, right? And you know that makes us different on that particular nucleotide. So we have a single nucleotide polymorphism at that location. People who are related would have more SNPs that are the same than people who are unrelated. Okay, and that has implication when we do things like, you know, DNA fingerprinting, which again we will learn about in lecture number 9. Moving on. Next one. Can someone tell me what the mutation is here? And we have the C here, and comparing this to the wild type, what kind of mutation is this? Okay. In the original, we have ACA, and now it becomes CCA, right? So threonine has now become proline. Okay, so this is a different amino acid, and Domain said it's missense. That is correct. This is a missense mutation. Okay. Now, is this bad or not bad? It's hard to say. Okay, it depends where it is happening in the entire sequence, and it depends what kind of proteins we're talking about. Okay, for example, if this is coding for an enzyme and it happens in the active site, it could change the shape of the active site, causing the enzyme to not work, and that could be bad. But if it happens elsewhere and the enzyme maybe is not as detrimental. Detrimental means like severe, as bad. Okay. Another thing to notice is we have 20 amino acids, right? And if you remember, like back to lecture 2, if you look at all the 20 amino acids, they are actually grouped together based on their properties. So some amino acids are like acidic, some are basic, some are going to be hydrophobic, for example. So if the missense mutations replaces an acidic amino acid with an other acidic amino acid, then maybe it's okay. Maybe it's not as bad. But if you replace it with a basic, replace an acid one with a basic amino acid, then it could have a drastic effect, right? So don't think of the mutations themselves as good or bad, but rather think of like where it is happening, right? What kind of protein is it happening to, and whether the replacement is actually of a same category or a different category in terms of the type of amino acids. I hope that makes sense. Okay, last one here. You know, we place the instead of AAC in the second one, it becomes UAC, UAA rather, and that is really bad because UAA is a stop codon, okay, and that is a nonsense mutation. Okay, and it creates a truncated protein. Okay, the rest of it is not even being made because there is a stop codon right here. Are we together? I think the A was also changed, right, in this one, if I'm not mistaken. Let's see. Yeah, so AAC becomes like UAC. So this is like a double base pair substitution. All right, let's take a look at here on the right side. We have base pair insertion or deletion. So this is where you put in extra nucleotides, you take out nucleotides, and that's going to cause, quite a bit of trouble. But again, the severity of the problem depends on where it's happening. If it's happening at the end, maybe it's okay. If it's happening at the beginning, could be a problem. Okay, so over here we have a deletion, right? Deletion. Okay, so deletion. And what happens is everything before the deletion, you can see, did not change, right? Same thing, right? Same thing, no changes. But afterwards, right? Because you're deleting this one, instead of reading ACA, right? Now it is reading like CAU and then AA something, something, something. So not only will you miss the stop codon in the original place, but you will be having wrong amino acids, everything downstream, okay? So this is a deletion that results in what we call frame shift. Okay, the reading frame has been moved, okay? And that results in extensive missense. Okay. This one, you have an insertion of a U. Okay. So insertion. And by inserting a U, you created a stop codon. So that results in nonsense. Okay. And you have a truncated protein. So if you're going to have an insertion or a deletion mutation, it's better to have a deletion or insertion that is a multiple of 3, okay? Because that will not change your reading frame. When nucleotides are inserted or deleted as multiples of 3, no frame shift will occur. Okay, so if you remove 3 nucleotides, worst thing that's going to happen is you lose 1 amino acid. That is probably better than having extensive missense. Okay. All right, that has been an hour, if you can believe it. We will take a short break, and when we come back, we will we'll create our own mutations. All right, so see you in 5. Okay, let's keep going. I'm gonna need a codon chart for the next part as well. Over here, oops, way too big. So let's see. The following is a template DNA of a section of a gene, and we need to create base substitution that results in neutral mutation. Right? So it doesn't really matter which part you pick, and there are multiple possible answers, obviously. So I'm just going to show you how to do it one way, and then maybe you can try to do another way. Okay, so first of all, this is template DNA. So what I need to do is I need to have mRNA, right? Because the codon chart here is based on mRNA, right? And you don't have to transcribe the whole thing. You can just pick anyone. Okay, so let's say I pick this one, right? Pick the second one. So if it's template CAG, then on mRNA, it is going to be GUC. Okay, GUC. So I'm going to look up GUC over here. GUC. And you see that GUC right here, GUC codes for valine, right? VAL. Okay. So I'm looking for neutral mutation. The meaning of neutral mutation is that you do not change the amino acid, right? So luckily you can see all these things here, they code for the same thing, VAL. So I can change the last one to any of the other nucleotides, and it will not affect what it codes for. Okay, so if we change GUC, let's say GUU, it's going to be the same thing. So there I've created a neutral mutation. But I must reflect that mutation in my original DNA, right? So if the messenger RNA is a U, then the original must have been an A. So if CAG got mutated to CAA, then it's going to be a neutral mutation. Okay, I'm gonna pick another one here. Okay, let's say we have CTC. Okay, CTC. What would be another neutral mutation? All right, I'll give you a moment to think about that, and let me know when you have an answer. Okay, so we have one answer from Tyler, CTT. Anyone else? Do you agree? Do you have something different? Remember, there are more than one possible answer sometimes, not always, but sometimes. Anyone else? Okay, so let me explain how to get the answer. So first of all, I need to make the mRNA, right? So CTC, the corresponding mRNA is going to be GAG. And if we look up GAG, it is coding for GLU, okay, glutamic acid. And we want another GAU, right? You can see both GAG and GAA codes for the same thing. So if I change this to an A, it's going to be the same. But I must reflect this change in the original DNA. So complementary to A is T. So Tyler is correct. This is CTT. Okay. All right, that's neutral mutation. Let's do the next one. Create a base substitution that will result in a nonsense mutation. So let's write down all the stop codons first. Stop codons. So again, stop codon is UGA, UAA, and UAG. Remember, these are RNA. So what would stop codon look like on a DNA sequence? Okay, especially template DNA, right? Okay, so on the template DNA, it is going to be ACT complementary, right? ATT and ATC. Okay, so that's going to be what a stop codon looks like on the template DNA. In other words, in order for me to create a nonsense mutation, I need to have one of these things in my DNA. So where can we do that? Right? We're just going to look for a place where we can create this thing. So for example, over here, right, we have AAT, but if I change the AAT to ATT, then it becomes one of the stop codons, right? When you translate it, transcribe it, sorry. And as a result, I have a nonsense mutation. Okay. Is that the only place for a nonsense mutation? Can anyone tell me another location where I can create another nonsense mutation? Are you guys with me? I am just a little bit concerned because you are all very quiet. Do you understand what's going on with this question? Okay. Thank you. All right, I will tell you another location. Okay, sure, I can explain again. So nonsense mutation means we have to create a stop codon before it's meant to stop, right? So normally the stop codon occurs at the very end, right? But if you have a mutation that causes stop codon to exist in the middle of the sequence, then you stop making the rest of the protein, right? Okay, so we call that a nonsense mutation. It creates a shortened or a truncated protein. Okay, so what happens is, in order to have a nonsense mutation, we need to have a stop codon in the middle. Okay, and we know that these are the stop codons, right? Okay, as they appear on the messenger RNA. Okay, but because we're looking at the DNA right here, the template DNA specifically, we want to transcribe these stop codons from the mRNA into the DNA form, okay, by using just base pairing rule. So, that's how I come up with these things. So I need to create an ACT, an ATT, or an ATC in the sequence so that I have a nonsense mutation.
What are we talking about in it though?
So another place where we can do it is here. You can see the CTC, right? CTC. If I change the first one to A, then I get the ATC, right? Which is a stop codon. And that would create a nonsense mutation. Okay. Hope that helps. All right, next one. Create an insertion mutation that results in a nonsense mutation. So again, nonsense means you need a stop codon, but this time, instead of a base substitution, I need to have an insertion mutation. I'm gonna have to put in something, okay? So where can we put in something? Well, for example, over here, we have the TTA, right? So what if I put an A here? If I put an A here, then the ribosome will read the codes, normally, right? TAC, CAG, GAG, AAT, CTC. But when it gets to here, because of the extra insertion, instead of reading TTA next, it will be reading ATT. But ATT is a stop, right? So the translation will stop right here, and the rest of it will not be made. Okay. Is there another location? What about this one? Can I do this? Can I put a T right here and create a nonsense mutation? Because now if I put a T here, I would have ATT, right? Is that going to work? Let's do a poll, okay? The 8 of you who were polling anyways, okay? Let's see what you guys think. My question is, when I put a T here, will that create a nonsense mutation? A for yes, B for no, okay? Because after I put in a T, I would have ATT, right? One of the stop codons that I have. So is that a correct answer or not a correct answer? A for yes, B for no. Oh, I have more response this time. Very good. Well, the answer is no, it's not going to work because again, right, let's think about the ribosome. What's the ribosome going to do? It's going to read it as TAC, CAG, GAG, and then AAT, right? It's not going to skip one and read it as ATT, right? It only reads in triplets, right? So it's going to read it as AAT. And then the next thing it will read is TCT, right? And then CTT, so on and so forth. So by putting it at the end is actually not going to create a stop codon. Okay, how about here? What if I put it, hold on, I'm trying to find if there's another place I can put this in. What if I put it right here? Is it okay if I put it right here? Is that going to be a nonsense mutation? Okay, let's try that one more time. A for yes, B for no. Okay, yeah. Almost all of you said it is yes, which is correct, right? Because ignoring this one, it'll read it as triplet, triplet, so on and so forth. And when it gets to here, instead of ATG, it will read it as ATC, right? Okay. So again, more than one possible answers, but you just got to be careful where you put it. Okay, last one. Create a base substitution that results in a missense mutation. This is probably the easiest to create missense mutation, because if you change the first nucleotide, you almost always end up with a missense mutation. Okay, so let's just take this one, for example, the AAT, right? And the messenger RNA codon will be UUA. Okay, so if I change UUA, let's just change it to like, C or something. Okay, then I will change it from, okay, what is UUA? UUA is LEU. Okay, and if I change it to CUA, oh, what is the chance of that? That actually did not work. That is just very unlucky for us. We will change it to something else. We'll change it to like, G or something. Okay, which means the original is going to be C. So GUA was GUA, right? It's going to be VAL. Right? So you change it from one to the other, it's going to be a missense. Okay. Let's do another poll question. Where are my poll questions? Okay, right here. All right. Let's try this. Very wordy. Go ahead. Okay. So if you delete this T, right? Then instead of reading TAC for the first one, then it will be like ACC, and everything is basically wrong, right? From that point on. So the correct answer is all the codons after that point will be changed. I'm not sure why people choose B though. RNA polymerase will correct the deletion and the normal. No, RNA polymerase cannot do corrections at all. Okay, so again, I'm not sure why people chose that, but no corrections can be made. Okay, what else is there to do? Oh, this is not bad either. Let's try this. Sharing, relaunch the poll. Okay, anyone else before I take it up? All right, so I have people choosing everything. And looks like D is the popular choice. Let's go through it. Okay, deletion of AAA at the beginning of a gene. Okay, what is that gonna do? What is that gonna do to your protein? Well, if you delete AAA, you will lose one amino acid, right? Whatever AAA codes for, you will lose that. Okay, it could be bad, but let's take a look at the other one. The codon UAC is mutated into UAU. Okay, so we can take a quick check here. UAC is the tyrosine, and UAU is the same thing. Okay, so over here, this is going to be a neutral mutation. Okay, so least harmful probably. And then we're going to have the next one. UAC is mutated into UAG towards the end of gene. If you remember UAG, right, you are good. That's one of the stop codon. So this is going to be a nonsense mutation. However, it's happening at the end of the gene. Okay, so like you're not going to lose like half of your protein. Okay, you lose a little bit. It could still be bad. Okay, but we're looking for the worst one, right? Okay, so let's keep looking. Deletion of A at the beginning of a gene. If you delete something at the beginning of a gene, then you're going to have a frame shift mutation, and that's going to cause extensive missense, right? In other words, you're going to be basically making a completely different protein, right? Not going to be functional for sure. And you know, that's going to be the worst one. Okay, and the last one, insertion of UAA at the end of a gene. UAA is a stop codon, but again, you're doing it at the end of the gene, right? So probably it's not going to be too bad. Definitely not as bad as this one. Okay, I'm not saying that, you know, none of these are bad. They are probably all going to have some kind of effect, except for the second one because it's neutral. But we're looking for the worst one, right? So this is definitely the worst one. Is that okay? All right. This one really gets people. Okay, this one usually people get it wrong if you are not being careful. Let's take a look. Let's see what happens. Are you gonna need a codon chart for this? Okay, anyone else? Has been 1 minute. All right, so let's see. Particular triplet has coding sequence of DNA AAA, the anti-codon on the tRNA that binds to mRNA. Oh, sorry, this is not the question I was thinking about. The one I was thinking about is something else. This one is straightforward. Okay, it is going to be UUU. Okay, because the coding and the anti-RNA, if you look at the cheat sheet that we had, right, it tells you what it is, right? Where is it? Okay, so the coding and the anti-RNA, they are complementary. So the correct answer was C. The one I was thinking of was this one. Like I would say the anti-code. Let's say there's a transfer RNA. Okay, and then this is like UUU or something. Okay, what is the identity of the amino acid? Okay, can you guys do that one for me, please? Okay, let me copy this strange looking thing, and then I'll put it right here. Okay, so what amino acid would it be? So let me give you some choices. Okay, is it A, PHE? Is it B, LYS? Is it C, ILE, or is it D, LEU? Okay, so please try this. Yeah, again, most of you got it. You don't actually look up UUU, right? Because this is the anticodon. So what does the anticodon bind to? Well, it will bind to AAA on the messenger RNA, right? Okay, so you actually look up AAA and the answer is going to be LYS. Okay, so the people who chose A, I hope I've convinced you why this is the correct answer. Okay, back to the sheet here. This one we have to oh boy, this is like a really long sheet. We're gonna have to identify the mutation. Okay, the original DNA shown is the template strand. It's very important that you know whether it's template or coding, because it would change completely how you do the question. Right. So in something like this, I mean, you're not gonna get something like this on the test necessarily, because it's not written, right? But like in a classroom setting, you probably would have to know how to do this. But I recommend chopping it into like triplets first. Okay, so we can see them clearly. And we're going to write the mRNA sequence. Okay, so the mRNA sequence is going to be AUG. Okay, I'm going to chop it, and then UGG, AAC, CGC, UGC, and UGA. Okay, it's just complementary, right, between the template and the messenger RNA. And we're going to look these up using the template, using the codon chart. Okay, so for the interest of time, I'm going to write it down for you. I'll post this whole thing online as well afterwards, so you guys can work it out on your own and see if you can figure it out. And if you're stuck, if you're not sure what's going on, feel free to let me know. Email me. I'll tell you. I'll answer your question. So that's the wild type. That's the original. Okay, and these exercises ask you to figure out what's wrong with the mutated sequence. Okay, so the first sequence here, I'm just gonna again chop them up into triplets. Okay, always do that first. Okay, so I see that it is the same number of triplets, codons, as the original. So that tells me I did not lose or gain any nucleotide. So it's not an insertion, it's not a deletion. It's probably a base substitution. Then I compare the nucleotides one by one. And surely enough, you will see that the C has been replaced with a T, right? Okay, so that is the changes. Okay, it's asking us to circle the change. There, we circled the change. So everything else is going to be the same. I'm not going to rewrite everything, but the second one, right? Second one is going to be, instead of UGG, we'll now have UAG, right? UAG. And you know what? I'm just going to write the first one again. It's not going to hurt. AUG, right, for the first one. And then now we're going to have UAG. And then the rest are the same. Okay? So if I look it up, AUG is MET. What about UAG? What is UAG? Anybody know what UAG is? You should know without looking it up, because I've been saying it all morning. UAG is stop. That's right. So will there likely be an effect? Definitely, it's going to have an effect. You're stopping on the second one. The whole protein is essentially gone. Okay, so what kind of mutation is this? Nonsense. Okay, that's all you have to do. All right, so I'm gonna try the next one with you again. Okay, but you should do it on your own, right, to figure it out. Right? It's much better that way. So you chop it up into triplets, codons, and you're like, oh, there was an extra one, right? That wasn't there before. So somewhere along the line, you probably have inserted something. and you have to find it out. So you match it, right? So again, TAC, that's fine. ACC, but now I see GAC, right? Well, that G is not supposed to be there, right? That's the insertion. Okay, so that's an insertion mutation. And as you can see, after you insert the G, everything is changed after that, right? So you will have a missense mutation, a frame shift. Okay? Insertion that results in a frame shift, which leads to extensive missense mutation. Okay, is there going to be an effect? Definitely, okay, because you're making a completely different protein than the original. Okay, so you should try the other ones. I'll post the answer after class, so you will have something to compare it to. All right. Long answer question. I'll quickly talk about it. So there are 64 codons, but only 20 amino acids. Explain why it is beneficial to have more codons than amino acids. This is basically an insurance policy, right? It's like a safety mechanism. And you can see on the codon chart that, for example, if you mutate the last codon, if you mutate the last nucleotide on these codons, you will end up having the same amino acid, right? So that's a neutral mutation. Right? So by having more codons than amino acid, you create redundancy. Okay, you create backups. So even if there is a little bit of mutation, that's okay. Right? You will not necessarily result in a harmful mutation. That's one way of looking at it. And another way is efficiency. Okay. There's something that we did not really talk about. And so, you don't have to know, but I'm just telling you. There's something called a wobble effect. You actually do not have you actually do not have 64 different transfer RNA. You don't actually have 4 different transfer RNA for each of these codons. There are maybe like 1 or 2 transfer RNA, because it turns out the binding of the last nucleotide, like this last one, that doesn't have to be a perfect match. Okay, so it's a little bit wobbling, right? We say so. It's called a wobble effect. So that way we reduce the number of tRNAs we need to transfer the amino acid. Okay, so for redundancy, safety purposes, and to minimize the number of tRNA necessary for the whole process. That's the reason. Explain which type of mutation is more harmful, more deleterious. Is it point mutation? Is it insertion, deletion? Again, we can't really say which one is worse. I mean, in general, if I have to, make a decision, I would say insertion and deletion is generally worse than point mutation. But does that mean point mutation cannot be worse than insertion or deletion? Absolutely not. Okay. If the point mutation occurs at a critical location in the enzyme, for example, that could completely render it non-functional, then that's pretty bad. Right? We talk about hemoglobin. It's a single point mutation, right? That causes the red cell to become sickle cell shape. That is really, really bad, right? Compared to an insertion or a deletion that's happening towards the end or something. It definitely is worse. So the key thing I'm trying to say here is, it's not necessarily the type of mutations that's more harmful or less harmful. It's where it's happening and how it is affecting the shape of the proteins in the end. Okay, application question. Deduce with reason what process is shown in each of the micrograph below. So the first one here. Anybody want to take a brave guess of what this is? Okay. Is it transcription? Is it translation? Or is it DNA replication? Let's start with this one. This one is probably the easiest. Okay. What do you think this one is? Is it transcription, translation, or DNA replication? Okay, we have transcription. Anyone else? Transcription. Okay. Yeah, I can see why you would think that way, but it's actually DNA replication. Okay. And the giveaway sign is the replication bubble here. Okay. So you remember last time we will have the helicase here, unwounding it. We have another helicase, unwounding it here, right? Okay. And so that is DNA replication. But you're like, well, don't you have to open up the DNA and you know, make a copy of it in transcription as well? Yeah, you do. But we don't really see the messenger RNA here, right? But over here we can see that you know, this little strand here is the DNA, and all these blue things that are flying out from it, those are actually the messenger RNA. Okay, so this one is the transcription. Okay, and I can hear you through this computer screen saying, but where is the DNA? I don't see the DNA, the one that is being copied. Probably it hasn't begun yet. Okay, remember, right? You got to open up the replication bubble. You got to have the single strand binding protein, and then the primase and all that stuff before the DNA polymerase comes in, right? So I'm guessing that this picture was taken before the RNA polymerase 3 came here, and that's why there's no DNA being replicated. You don't see it. Okay, and finally, this is translation. Okay, all these purple balls, those are the ribosomes. Okay, all these green ones that are coming out, the green little line, zoom in a little bit for you. All these green lines, those are the amino acids being linked together, the polypeptide. Okay, and you don't really see it clearly in this picture because all the ribosomes are kind of hugging the whole RNA, but you know, if I trace it out for you, you see the purple strand here. That's actually the messenger RNA. Okay. So I'm not gonna give you like a brand new picture that you haven't seen before, but I might give you one of these pictures on the test and ask you, you know, what is it that you're looking at, and you should be able to tell me what it is. Okay. Well, that is it. I'll post the answer for the fill in the blanks, and you can you know, look at it yourself, compare the answers. And that's it for this week. Okay. I really hope I'll see you again next week. You know, but if not, then you know, take care of yourself, and hopefully we'll meet soon again. Okay. And of course, you can still email me throughout to the end of the week. You know, if you have any questions, or if you choose to do the quiz a little bit earlier. Okay. All right, that's it. Thank you for coming. Take care. Bye now.